I'm Timothy Becker, and I'm presenting the paper, A Framework for Associating Structural Variants with Cell-Specific Transcription Factors and Histone Modifications in Defect Phenotypes by Becker, Barasan, and Shin. And this is for the workshop Data Mining from Genomic Variants and its Application to Genome-Wide Analysis, 2022. The motivation for our work was Cancer Genome Atlas and Kids First data portal, GWAS studies have generated high throughput short reads, the Illumina paired end data, and they've used it to do successful single nucleotide variation. But not as many of those studies have done structural variant analysis. The structural variants for SVs are known to affect more total base pairs, but they're difficult to discover, they have lower accuracy. One interesting thing that motivated our work was that they can often target genes indirectly through a cis regulatory function, an enhancer, a promoter, and so they can have this indirect relationship, which we wanted to explore. Another motivating factor was that older assays, such as array-based CGH, have had some defect associations. And one interesting side to this is that even with a total analysis of single nucleotide variants and structural variants, in other words, the total genomic, it's only the potential driver. There are epigenetic and environmental factors that are also known in many defects. Structural variants are the larger variants, so they're typically larger than 50 base pairs, and they have specific subtypes like deletion, duplication, insertion, inversion, and translocation. For the short reads, the Illumina paired reads, there's four main signal types, which are the read depth, read pair, split read, and assembly. And for emerging long reads, which are typically greater than 4,000 base pairs, the split reads and assembly reads are the typical signals. Most of the studies that are available now are the short read, and in our work, we're going to focus on those. The Gabriel Miller Kids First data portal has an oral facial data set that we're going to make use of in our study, and this is Illumina paired read, 125 base pairs, and it's greater than 25x coverage. The Gabriel Miller's Kid First and Cancer Genome Atlas have both made use of an existing annotation tool called Variant Effect Predictor, or VEEP, and this works really well for single nucleotide variants. But it's too general purpose, and there's a lot of defect-specific information that can be used. In our work, we made use of this literature, which gave us genes, pathways, transcription factors, and even cell types. And when we leverage all of this external information, it allows us to infer SV functional class. So our workflow is as follows. We look at the literature, and this gives us gene lists. From the genes in the gene list, we can associate pathways. Some of the genes in our gene list may actually be transcription factors, or they may be controlled by transcription factors. And finally, when you look at a particular defect like oral facial cleft or OFC, this is going to lead you to look at specific cell types like neural crest and mesenchymal stem cells. The other thing that we use in our workflow is a short read DNA seq. Typically it's DNA that we have, and we don't have RNA. We also are designing the workflow to work on an entire cohort. And the cohort, because it's defect specific, we often have a parent. So it's a duo or trio, and we have phenotypes for each. The child has the defect phenotype. Usually at least one parent doesn't, and if we have that, then we can utilize that information to filter out potential SVs. We then take all of that and we integrate it with external sources, like known SVs from the healthy population in 1,000 genomes, or 1 kg. We use histone modifications from ENCODE, and we use transcription factor binding sites from the Jasper database. Figure 1 shows the overall framework. We do our best to call and genotype the SVs, and these come in at the very left-hand bottom in panel A. We then filter out the healthy population. If we have parents that don't have the phenotype, we filter that further, and this gives us a set of novel SVs. We then pass those SVs over to panel B, which is the integration, and from that part we look at the gene landscape. This allows us to look at transcription factor binding sites that are inside histone peaks that are inside a structural variant. And this picture here in B allows us to make 5 distinct classes that you can see in panel C. We have the direct gene class. This is the SV is happening right in the region of a protein coding gene. We have poised enhancers, which they have a histone peak that's signifying poised enhancer. A slightly different histone mark would be an active enhancer. A promoter would need to actually have the correct histone modification, have transcription factor binding sites, and also be very close to a transcription start site or a TSS. Finally, if it's not poised, it's not an active enhancer, it's not a promoter, it's not direct, it's a intergenic SV, but if it's not any of the cis-regulatory ones, we define it as non-regulatory. So those are our 5 classes. To test our framework and develop it, we looked at the Gabriel Miller Kids First Oral Facial Cleft Asian and African trios. This was around 250 trios. The child has the phenotype, and typically most of the parents don't. We then derived a gene list from that exact same literature for the scientists that generated that DNA sequencing. We then pulled external histone modification data from ENCODE, and we pulled the transcription factor binding site files from the Jasper22 database, and we completed a comprehensive SV analysis with Fuser SV and Tensor SV. Overall, out of our 17 families, the children had more total SVs. So they had more total SVs, which is unusual. But again, they had the defect phenotype and all the parents didn't. When we dove further into it, we found that the children had the same amount of direct SVs. So there was not a lot of direct SV, and this is very rare. However, using our classification from the method in figure 1, we found it very interesting that the children had elevated regulatory classified SVs. So they had poised enhancers, they had active enhancers, they had a few promoters, and these were elevated on average more than any of the parents. So that's the overall analysis. We then completed an alternate analysis, which was looking at only the novel SVs. And so these were the ones that were interested. Are these novel SVs, are any of them associated with the OFC phenotype? And to do that, we kind of filtered out the parents. We also looked at the average intersection that gave us a picture of the SV inheritance between the parents. Parents on average had around 350 unique SVs to themselves that were not shared in the trio. The children had around 289, and this is because the children are inheriting from both parents. So when you filter out all those common SVs, the 289 is actually, like we saw, it's a little high. It should actually be less. When we plot using IGV a particular SV that we call that's in the child, such as figure 3 in the paper, we can see we have a tandem duplication, which is actually occurring on the FANCA gene. It looks like it's covering at least 3 of the exons in the FANCA gene, but that is also within range of enhancer, promoter, and active enhancer signatures for the mesenchymal stem cells, which are shown in purple. So this is an example of how the framework works. It's going to actually classify this tandem duplication as regulatory. If those histone modifications or transcription factors weren't present, then this would still potentially be affecting the FANCA gene. So this is kind of what we're looking at. This is just one of the samples, KFN 436. You can also see this structural variation is only in the child KFN 436, and it's not in the parents 437 and 438. The other interesting thing that we completed with the framework by looking at the cohort is we looked at common SV targets. So even if the SV wasn't overlapping, in other words, it wasn't the same SV because these SVs were occurring novel for each of the OFC phenotype children, we saw that they had some common targets that were being affected. Even though the SV is different, it's within range and it meets the criteria of being like a poised enhancer or an active enhancer. We found 6 interesting indirect regulatory targets. We found EPHB1. We found a poised enhancer that was deleted, so it was a potential poised enhancer, but it's now missing. And that was in 35% of the 17. We found FOXO3b had a duplicated poison enhancer in almost half. So almost half of them had some kind of poison enhancer that was duplicated in the FOXO3b region. We found almost half had an active enhancer that was duplicated in the MAFA region. We also found, very similarly, in the MAPK15, we found active enhancers that were duplicated in and around the MAPK15 gene. We found poison enhancers in around 1/3 that were deleted for Reich, and we found even inverted SV types. There's an active enhancer that was inverted, and 41% had inversions in the THBS2 region, which is very interesting. So in summary, the SVs are more complex than SNVs in several ways. Calling them is more complex. Genotyping them is more difficult, and finally annotating or figuring out their function is more complex. The context requires defect-specific association tools. So what we're working on puts a lot of information together, and we're working on this as a Python 3 implementation. What we found is that by looking at the OFC data, we found that it's very much regulatory associated in literature. The non-syndromic form of oral facial cleft does not have a lot of direct gene targets, either in single nucleotide variants or in SVs. In fact, the single nucleotide variants in the existing literature are also regulatory. And so we found that in our pilot study that it supports existing OFC literature, that there is a lot of regulation-driven variation for the non-syndromic OFC phenotype. What we need to do to further our work is to complete all of this data set. So we would want to do all 759 samples before we were able to make our final conclusions. And of course, this would also allow us to refine our framework, maybe add additional outside sources of information so that we can provide a more total context of what's going on in the cohort. This completes the end of our talk, and I believe that we'll have questions that will be answered in person. Thank you.